Welcome to The Explainer. Today, we're going undercover. We're putting on our detective hats to investigate the totally invisible world of bacteria. Our mission? To figure out how in the world scientists can possibly identify these microscopic suspects. So let's start with a wild fact. Right now, about 30% of us are carrying a pretty notorious bacterium, Staphylococcus aureus, right in our noses. Yeah. For most people, it's just hanging out, not causing any trouble, but for some, it can become a really dangerous intruder. And that really brings us to the central mystery of our investigation today, doesn't it? When you literally can't see the culprit, how do you build a case? Well, it all starts with the very first clue. Okay, let's get into it. Every good detective needs a way to sort the suspects. And in the world of microbiology, that first step is a technique that's actually over 100 years old, but it is still absolutely fundamental. It's called the Gram stain. Think of the Gram stain like the very first forensic test you'd run at a crime scene. It immediately divides all your bacterial suspects into two very distinct groups, all based on the type of armor they're wearing, their cell wall. And here are the results. Gram-positive bacteria, they've got these thick cell walls that trap a purple dye, so they look purple. Gram-negatives, on the other hand, have thinner walls. They don't hold on to that first dye, so they get stained pink instead. Just like that, you've cut your suspect pool in half. It's their first big tell.
So let's open up the case file on one of our most wanted gram-positives, the very same one we found hanging out in our noses, Staphylococcus aureus.
And here it is, the complete rap sheet for S. aureus. You can see its aliases, what it looks like under the microscope, kind of like a cluster of purple grapes, and its usual hideouts. But that known crime section, well, that's where it really earns its reputation. This bug can go from being a totally harmless bystander to the mastermind behind some really serious conditions, including the infamous, hard-to-treat MRSA you hear about in hospitals. So the really crucial part is understanding how it pulls off these crimes. This is its arsenal, its virulence factors. It can create this slimy shield called a biofilm to stick to things like artificial joints. It can pump out powerful toxins that cause nasty food poisoning or even toxic shock. And it can even produce enzymes that literally chop up our antibiotics. Now let's look at the other side of the lineup. Time to turn our investigation to those pink-staining gram-negatives and profile another household name, Escherichia coli. Come on, we've all heard of E. coli. And as this file shows, it's a totally normal resident of our gut. Most strains are actually an important part of our body's whole ecosystem. But this case file also shows that certain rogue strains are behind a huge number of infections, everything from a common UTI to really life-threatening conditions like meningitis in babies or severe kidney damage. And this is where things can get really serious. Strains like the notorious O157H7, which you often hear about in food poisoning outbreaks from undercooked meat, have this devastating secret weapon, something called the Shiga toxin. This thing can literally shred the lining of your intestine and cause severe kidney failure. It's a perfect example of how one particular strain can become a major threat to public health. You know, just when you think you've got it all figured out, you've sorted your suspects into purple ones and pink ones, the microbial world throws a few curveballs your way. Some of these bacteria, they just don't play by the rules. Yeah, these are the outliers, the masters of disguise that you just can't identify with our standard gram stain lineup. They need their own special investigative techniques. Okay, check out these three. Mycobacterium tuberculosis basically wears a waxy raincoat that makes it invisible to our gram stain. Then you have spirochetes, which are like these tiny thin corkscrews that just slip by undetected. And then there's chlamydia, the ultimate stealth agent. It does all its dirty work from the inside by hijacking our very own cells just to survive. And this brings us to the most urgent part of our investigation. The suspects, they aren't just sitting still. They are constantly evolving. They're actively fighting back against our best weapons, which has created a massive global crisis of antibiotic resistance. So why does this matter so much? Because bacteria are absolute masters of survival. As this explains, they've come up with an incredible range of counter-espionage tactics. This isn't just a science problem anymore. This is what makes a routine infection suddenly become life-threatening. It's what complicates everything from major surgery to chemotherapy when our frontline drugs just stop working. This whole process is just a stunning illustration of evolution in action. Think about it like this. When we use an antibiotic, it's like a police raid. It wipes out most of the bad guys. But if any of them have a random mutation that makes them resistant, they survive. And with all the competition gone, these superbugs are free to multiply and completely take over. And get this, they can even share their resistance genes with other bacteria. And here are their top 3 strategies. First, they can produce enzymes that act like scissors, just chopping our antibiotics to pieces. Second, they can change their own structure so the antibiotic's key doesn't fit the lock anymore. Or third, they can install these tiny little pumps in their cell walls that actively push the drug out just as fast as it comes in. It's a full-blown biological arms race. From a simple stain that tells us purple or pink, all the way to the insane complexities of antibiotic resistance, our detective work reveals a world that is just constantly changing. The story of bacteria is one of incredible adaptation and survival. Which, of course, leaves us with one critical question. As they continue to evolve, how will we adapt our methods to stay one step ahead in this investigation?